I'm Nadine Dagan, CEO of Hearing Health Foundation. I wanted to thank you all for joining us today for this special research update by our Hearing Restoration Project Scientific Director, Dr. Peter Barr-Gillespie. A little background on me. I joined HHF in January as the CEO, but had previously consulted with the Foundation prior to my accepting the current position. I have a very personal connection to HHF's mission. My father has adult onset tinnitus, which is often debilitating. Sorry, I apologize for the sirens in the background. We are in New York City. And my brother has had hearing loss since childhood. My youngest daughter, now 5, had painful reoccurring ear infections when she was a baby and toddler that had also led to residual hearing loss. So I am personally motivated to find better therapies and cures to enhance the lives of my loved ones as well as the millions of individuals living with hearing loss and tinnitus. Joining us today for the live presentations are the members of the board of HHF, the staff, and other hearing restoration project scientists, as well as longtime supporters. We appreciate and thank you for your continued support and interest in groundbreaking research. Our goal is that everyone who listens to this presentation will obtain new information on hearing loss and our research towards cures and treatments. If after listening to the briefing, time passes and you have questions or thoughts, are your source for reliable, up-to-date information. We intend to have more webinars with our researchers in the future. Thank you again for your time and interest in the Hearing Restoration Project. For those of you who have just dialed in, we ask that everyone mute their phones by pressing *6. There will be opportunity at the end of the presentation for questions, but during Dr. Peter Barr-Gillespie's presentation, we ask that all lines be muted to minimize audio interference. Please feel free to type your questions into the chat box. Thank you for your cooperation in joining today. Brief history on the Hearing Health Foundation. We were founded in 1958, almost 60 years ago, and we have established a reputation for pioneering breakthroughs in hearing and balance research. Some examples of this include we were early supporters of the revolutionary cochlear implant, and today over 220,000 children and adults are benefiting from this. We also advocated for the passage of the universal newborn screening legislation in the 1990s, and now over 97% of newborns are tested for hearing loss at birth. Our emerging research grant program has and continues to provide seed funding for early-stage scientists in hearing and balance science. Many of our funded researchers have gone on to obtain federal funding for their work, and this is one important measure of success. Experience throughout the years leads us to the focus today, the Hearing Restoration Project Consortium. This consortium is tasked with finding and developing effective therapies for hearing loss and tinnitus through hair cell regeneration. I would now like to introduce Dr. Peter Barr-Gillespie, Hearing Restoration Project Scientific Director and Professor of Otolaryngology at Vallon Institute at Oregon Health Science University. Dr. Barr-Gillespie will now present on the HRP's plans to develop effective therapies for hearing loss and tinnitus. Again, please mute your lines by pressing *6. There will be an opportunity to ask questions at the end. Thank you.
Thank you, Nadine. And we go from noisy New York to quiet Oregon. I thought I would give you first a quick overview of what my presentation is going to encompass. I'm going to first talk about a new member of the HRP consortium. I'll then give you a refresher on what the HRP is doing and how we're going about it. Next, I thought it would be of interest to present a year in the life of the HRP, how the investigators get together and how we discuss projects and develop projects. I'll go on next to giving a couple of vignettes about some ongoing science that's very exciting, and then conclude with a brief discussion of our plans for 2016 and beyond. So the first personnel update is first. I'd just like to announce to those of you who don't know that we have a new HRP member, Ronna Hertzano from the University of Maryland. And Ronna is a clinician scientist, which is a rare type of person and a really important one for us as well. So she has a clinical practice, a surgical practice that focuses on diseases of the ear and the lateral skull base. But she has a very active research program both into hearing restoration, genetic causes of hearing loss, and mechanisms of control of genes that are important for hearing. And importantly, she's developed a platform, a bioinformatics platform called GEAR, which allows visualization of large genomics data sets, which the HRP generates. And just to give you a brief flavor of the GEAR, which is the abbreviation for a portal for gene expression in auditory research. I give you this slide here. Don't want to worry about the details, but the common thread here is that the complex genomics data sets that we have that may have 20,000 different entries can be distilled down to single entries that can be compared across different data sets. And those of you who know about the HRP know that we're generating data sets from the mouse, for example, one here, the fish, like the zebrafish, one here, and the chick, like this one here. And this visualization platform allows us to compare the results for a single gene. Here is the example of a gene called SOX2. We can compare the results across these data sets and maybe learn about the similarities and differences between the different species and model systems that we're using. So we're really excited to have Ronna on board. I think then I'd like to talk briefly about the HRP as a whole, just to remind you of what we're doing. This was founded now almost 5 years ago, and the HRP consists of 14 hair cell regeneration investigators, plus me as the scientific director. And we do things differently. We actively collaborate, share our data, and share our ideas. And this allows us to make progress more quickly than would occur when each individual investigator is locked up in their home department at their home university. So we cross those barriers between institutions and allow for collaborative research. The HRP funds projects that are not traditionally funded by the NIH. For example, those large genomics data sets that I alluded to. And finally, we have a scientific advisory board made of scientists in our field who are not part of the HRP consortium, and they provide an oversight role and also provide guidance to our consortium. So as you all know, the sensory cells of the ear are located deep within the internal ear, and here is the internal ear right here. And there's two main components. There's the vestibular system, which gives us our sense of balance, and there's the auditory system, which gives us our sense of sound. And the cochlea, which is this snail-shaped structure, is responsible for detecting sound. Most of you also already know that the sensory cells within the cochlea or the vestibular system are called hair cells, and they're the key cells in the auditory and vestibular system. They detect sound or head movements and convert it into electrical signals in the brain. So here is a picture of the cochlea. Each of these white spots that you see here are the sensory organelle of the hair cell, which is called the hair bundle. Here's some here, here's some here. And this is a normal cochlea. This is what you want your cochlea to look like. Unfortunately, time, age, and noise exposure, and also exposure to ototoxic drugs, which are drugs that kill hair cells, all of these will then lead to damage to the hair cells in the cochlea, leading to a situation like this. And the fewer the cells, the less functional the auditory system is. So the HRP is taking advantage of information that we've had for some time, that there are species that show robust hair cell regeneration and those that don't. Unfortunately, humans don't show hair cell regeneration, at least in their cochleas. However, the chick and the fish, the zebrafish is our favorite fish to study, show robust hair cell regeneration after the hair cells are damaged. But like humans, mice don't show hair cell regeneration, at least in their cochlea, in their auditory system. They show limited regeneration in the vestibular system, which we also take advantage of. So the idea is to compare these three model systems. Now, hair cells are shown diagrammatically here, surrounded by a variety of classes of so-called supporting cells. And what damage does to the hair cells is causes them to be obliterated from the cochlear epithelium. In some cases, the epithelium repairs itself, and in other cases, the epithelium degenerates. But in either case, there are no functional hair cells. And the goal of the HRP then is to go from either this repaired epithelium or this flat epithelium back to the situation with hair cells. And we need to stimulate regeneration pathways, sequences of molecules that will cause hair cells to appear, or we need to inhibit pathways that block regeneration in the cochlea. And so we're trying to understand which of those approaches is what we need to do. The strategic research plan, which I'll refer to a couple of times here, has 3 phases in it. The first phase is to carry out discovery research where we find those so-called pro-regeneration or anti-regeneration pathways, and also to determine what happens in the cochlea after damage. And this requires a lot of discovery experiments where we just damage these preparations, damage their hair cells, and then look across the genome, across 20,000 different genes, to see what happens to the gene expression, either in an organism that shows regeneration or in an organism that doesn't. Once we've identified these candidate pathways, we move to phase 2, where we verify these pathways in the different model systems and prove whether or not the hypotheses that were generated in phase 1 are correct. In phase 2, we also develop assays that allow us to validate these pathways. Those assays likely can be adapted to screening for drugs that trigger hair cell regeneration in the mouse. And that would be phase 3 of the HRP's project. And we have been really working on phase 1 for the whole time of the HRP, and in the last several years have moved into phase 2, although we will continue to conduct research in phase 1 throughout the life of the project. One of the things that you learn in science is that the more you learn, the more there is that you don't understand. But at the same time, technological advances and advances in understanding of cell biology in other areas mean that every year our ability to determine what's going on at a molecular level gets better and better. So the discovery research will continue, but at the same time, our pathway validation, we're already moved into that. So that's the HRP's plan. How do we operate on a year-to-year basis? And there are a number of activities that we carry out, and I think it's worthwhile talking about each of them. First of all, monthly or sometimes more frequently, we have phone calls where we discuss projects and also share data using Adobe Connect, just like we're using right now. So at each meeting, 2 to 3 of the HRP members give brief presentations about their progress on their funded proposals. And then that leads almost invariably into vigorous discussion amongst the group, which will help the HRP members in deciding where to go next with their project, but also allows cross-fertilization between projects. There are also additional phone meetings among the different projects, many of the projects are multi-investigator and often multi-institution. And so HRP members are talking on the phone or over the internet on a very frequent basis. A key event in the HRP is the fall meeting. So this is a 2-day meeting typically held in Seattle where we get together on a Sunday and have a working dinner and then get into great detail on Monday about the activities of the HRP. This is crucial because all of us are brought together in the same room. We have a lot of time to discuss progress, discuss ideas, and come up with plans for the future. So on the Monday, which is the full day that we have for our meeting, we have collaborative meetings among the groups. We have research updates. But most of all, we review our progress and decide where we're going over the coming year. On the last day, we discuss new projects and figure out how they assimilate into the research plan. Both in our discussions on the Sunday and discussions on the Tuesday, we'll revise the strategic plan as needed. Coming out of the fall meeting, HRP investigators develop proposals for next year's funding. So the ideas are brewing at the meeting. Preliminary suggestions are discussed there and then between people by email or phone that then coalesce into one-page pre-proposals that all HRP members discuss. So this is a crucial step. We put our ideas down on paper and chew them between all of us. Some of the ideas end up getting dropped. Maybe they don't fit into the research plan. Other ideas are merged together, so we have a larger proposal of coordinated activities. So based on those discussions, each of the projects, which typically includes multiple investigators, each project would submit a full proposal, which is then evaluated by all of the HRP members who are not on that proposal. So everybody gives feedback and returns it to me, and I present the full proposals with the evaluations by the HRP, as well as my own evaluations. I present them to the Scientific Advisory Board, and the SAB does the actual evaluations. And they sometimes send some of the proposals back for more work, and then those investigators prepare rebuttals that are then evaluated by the SAB. So this step is a really important one. Having these outside scientists look objectively at our work, I think really helps us in making sure that we have the best possible science. We have a second meeting, which happens at a large meeting that almost all of the HRP members go to, the ARO meeting, which is held typically, and it's been rotating between San Diego and Baltimore. And we have a 4-hour dinner meeting either in San Diego or Baltimore, which is attended not only by the HRP members, but also by many of the scientific advisory boards. And this gives us additional opportunities to discuss HRP's goals and plans. And I think it's very important because it allows the Scientific Advisory Board to directly comment on where we're going. And it leads to plenty of unrestrained discussions amongst the HRP and amongst the HRP and the SAB. I'm going to just hold off a moment here. I just ask everybody to please mute your microphone on your phone, if you will, please. Thank you. So, as you know, we have funded a variety of projects in 2015, and these are wrapping up right now. I'm not going to talk about all of these in detail, but I will allude to a couple of them. Many of you know about a very exciting project called the Excel project, which was which was brought together by 6 of our investigators who jointly recognized that in the mouse there were cells that were seemingly responding to damage to hair cells in a way that suggested maybe supporting cells are turning further into hair cells than we thought. And so this was a very exciting result that came out of the 2014 fall meeting in Seattle and led to a proposal and a quick funding of a project to study these cells in more detail. And this is, of course, a good example of how science works. It was maybe 2 steps forward, 1 3/4 step back, in that the group was able to characterize these X cells more and find that while there were some very rare, interesting cells that we remain interested in, they were rare enough that it was difficult to characterize them. And other cells which showed characteristics of hair cells did not seem to be what we were looking for in terms of cells that were likely on their way to regenerating hair cells. So the X cell group is wrapping this project up. It's not dead. We're still thinking about what it means, but we haven't been able to take advantage of it in the way that we hoped. As you can see, there are a number of other phase 1 projects that are going on. The chick utricle response to damage and zebrafish single cell response projects I'm going to talk in brief about them a little bit later. I'm also going to allude to some of the work that's done in this cochlea project. But suffice it to say that we're making progress both in bioinformatic comparison of our data sets, also in characterizing another level of regulatory molecules called microRNAs. We'll know more about these soon in this project that is still ongoing. But this is a good example of how we're continuing to need to work on our phase 1 because there's a lot more to learn. That said, we have moved, as I said before, into phase 2 with a number of projects for 2015, including one that uses all 3 of our model systems to compare different pathways to see if there's similarities between fish and chick and differences with the mouse. And this project will continue on for 2016 as they've made significant progress and really have provided a couple of models for testing pathways. There are several other phase 2 projects that are ongoing, and there are some new ones that are going to be funded for 2016 as well. So in this part of the presentation, I'd like to switch to giving you a couple of vignettes about some ongoing work in the HRP. I'm going to give you 2 vignettes. The first one is work that's carried out by Neil Siegel and Andy Gross, asking the question, are regeneration genes blocked in the mouse? Are there genes that have had their structures changed so that they no longer work? If those structures were reverted back to a state where the genes could be turned on, maybe we could overcome a block to regeneration. This requires first a little bit of a refresher for basic biology. Remember that each of the 23 pairs of chromosomes that we have in the human has a variety of levels of structure that as you go from different scales, the structure changes in different ways. But when you get down to the level you see right here where the individual DNA strands, which are the blue strings, are there, you see that the DNA strands are wrapped around proteins called histones. And histones compact the DNA, but also control the access of the DNA to regulatory molecules which control the activity of the gene. Under certain circumstances, the gene's turned on. Other circumstances, the gene is turned off. And we can recognize those modifications by looking at directly at chemical modifications that the cell makes to the histone proteins. So Neil and Andy are interested in histone modifications, some of which turn off gene expression, some of which turn on gene expression. And they're also interested in the overall structure of the DNA around the genes of interest, because when the gene is actively turned on, the DNA is made much more accessible. And we have really remarkable technologies that allow us to look at both of these facets of the cell biology across the genome, across all 20,000 genes in the genome. And I'm not going to show you all 20,000 genes, fortunately, but I will show you one. And this is a really important gene called atonal 1, or ATO1. As many of you know, ATO1 is a master regulator of hair cell formation. Hair cell has to turn on ATO1 in order to become a hair cell. And certain cells in the ear can be forced into a hair cell fate by turning on their ATO1 gene. So it's a very important master regulator. And here is the linear stretch of the gene itself. And this is the part that controls the protein ATO1, and this is the part that controls how that protein is turned on and off. And I won't go through the details here, but suffice to say that you can look at a histone modification like this one right here and see that in hair cells, this histone modification is turned on to a much greater degree than in supporting cells, which fits with this gene, ATO1, being turned on in hair cells and not in supporting cells. And likewise, this technique reveals the openness of the DNA, and you can see it's greater, there's greater access to the DNA in hair cells than in supporting cells. So we have this information on all the genes of the genome. And the next question, and it's the big one, is to ask which of these genes, particularly ones that are turned on or that are known to be important in supporting cells, which of the genes have their epigenetic marks, as we call them, changed as mice lose their ability to regenerate hair cells. We're taking advantage of the fact that immediately after birth, at postnatal day 1, mice have the ability to have hair cell regeneration in their cochleas, but within 5 or 6 days, that ability is lost. And so if we compare the epigenetic marks at the early and late stages, we may learn which genes are specifically turned off in supporting cells, which may be the key genes to control. If we see genes that are turned off, the next question will be, can we reverse those changes? And if we do, will that allow hair cell regeneration? Those are big and challenging questions to address, but this is a really important step because this control of gene expression may underlie the refractoriness of mouse and human genes to, or cochleas to hair cell regeneration. So in the next vignette, I'm going to just present a little bit on how we're getting better resolution of events from analyzing single cells rather than large groups of cells in the ear. This is work that's carried out by Stefan Heller and Tatiana Piotrowski. Stefan's largely working on the chick and Tatiana on the zebrafish, and they're carrying out very similar experiments in collaboration. And what they're doing is taking advantage of some modern technology, which is spectacular. It allows you to take an organ, like the chick utricle, but it could be the fish, zebrafish ear, or what have you, then dissociate all of the cells into individual cells. Sorting machines allow us to pick out single cells, and then advanced technologies allow us to carry out an analysis of what genes are expressed in each of hundreds or even thousands of cells. And the pattern of expression of those genes in those individual cells tells us something about what that cell has been doing recently. So when you analyze all the cells, this is several hundred cells from a chick vestibular organ, you analyze their gene expression, you get figures that look like these, and let's not worry about the details. But let's particularly look at this figure here on the left. This figure allows us to see the similarities between different cells. Each of the dots corresponds to a single cell. And you can see there are a bunch of cells that are similar to each other here, and a bunch of cells that are similar to each other here. And other analyses show that these are hair cells and these are supporting cells. So what this is saying is that the gene expression patterns of these two types of cells are very different. So the key experiments in these single cell approaches, is to carry out damage to the organ where the hair cells are killed. And in the case of the chick experiments, the damage is aminoglycoside drugs, which kill the hair cells. And here you see a similar experiment to the one that was shown in the previous slide, but it's one mixing cells before the damage and cells after the damage. So the cells before the damage are in orange and the cells after the damage are in blue. And you can see that most of the cells overlap, that there again are hair cell cluster. Now it's in a different place, but that's not significant. And another supporting cell cluster. But there's some real interesting new clusters that appear from these damaged ears. And one of them I'll just call your attention to right here. Most of its gene expression pattern of these cells is similar to supporting cells. But specific genes, like this one here, are known to be hair cell specific genes. And what this seems to be saying is that this is a population that is on its way from turning into a hair cell from being a supporting cell. At least that's our early interpretation. And there's a lot more data yet to be collected in these chick experiments, and very similar experiments are being done in the fish. But it's very exciting because it allows for resolution of changes in gene expression in cells very soon after damage, and presumably we'll be able to follow during the whole hair cell regeneration process how the gene expression changes. So that's the big question. We want to know what happens immediately after hair cells are damaged, before they die, and then what happens early on in supporting cells. And we will first do all this in the chick and the fish because we know they regenerate hair cells. But the key question is, which of these events that we'll catalog fail to occur in mice? So we'll carry out similar experiments in mice as well. The genes that are not undergoing the transitions that in the mouse that we see in the chick and the fish are then potentially targets for manipulation. So where are we going next? I want to emphasize again that phase 1 will continue as technologies evolve and we learn more from other areas of science. I mean, that's one of the beauties of science, is everything builds on everything else. That said, we've got active phase 2 projects going on now. We've got some exciting new results in the chick and in the fish using gene knockout experiments in the fish using something you may have heard of, which is the CRISPR gene modification method. And we also have experiments in the chick using inhibitors of pathways that will allow us to find out, at least broadly speaking, what's important for regulating regeneration. We are now moving to scaling up phase 2 because we need to be able to screen many genes because many candidates will come out of phase 1, and we need to be able to screen them in combination. We've got a project that will be funded for 2016 that will develop cell lines from the ear that will allow us to screen for drugs that can cause supporting cells to divide and grow, or even to turn into hair cells. And then in the future still is the drug screening step. But I think, as I've mentioned before, the screening model is really, really important. And that is something that we're actively working on in phase 2. So in summary, I would say that triggering hair cell regeneration remains a very promising strategy. It's, of course, more challenging than we thought it was starting out because the system is more complex than we thought. But I think we're making significant progress. The advancements in technologies, not necessarily developed by us, but developed by others, means that we have a greater and greater ability to study hair cell regeneration in the different model organisms. And finally, I think that the comparison between the model organisms and the sharing of ideas and many other facets of the collaborative interaction model mean that this has been already a fruitful approach and will remain so for the future. So in summary, I'd say that it is correct to say, the question is not if we're going to be able to regenerate hair cells. I think that's quite likely. The question is just when. We've made a lot of progress, and I think it's just a matter of time and investment in our model, our collaborative model, and in other ways of studying hair cell regeneration. So I'll conclude by handing it back over to Nadine.
Thank you, Peter. We appreciate your work and dedication to HRP and for providing all of us with this wonderful update. I also want to thank Laura and Veronica here at HH's office for coordinating the logistics so we could hear your briefing. So I'd like to follow up to see if there's any questions in the chat. Can you move to this? Yeah. Is there anybody with any questions? You can unmute your phone by hitting *6 again. Okay, it doesn't look like there's questions, which I guess means you did a great job, Peter. So our goal today is to allow everyone to obtain new information about hearing loss, research towards a cure, progress to date, and our outlook going forward. Oh, there's someone typing, apparently. That's exciting. Peter, can you see that?
Yeah, Kate says a lot to process here, would like to review the recording, which makes sense. I am certainly available at any time.
I think I'll have more questions when I let all of the things I just saw sink into my brain for a bit, because I feel like this one was maybe a few steps above technically the first one, and so I want to settle into it and process all of it, but I think at the moment I haven't processed it enough to come up with an intelligent question.
No worries.
We'll be sending the link with the entire briefing.
I'm available for any questions too.
Okay, so like I said, we'll send out the briefing to everyone who participated so you can review it again at your leisure. And please feel free to reach out to myself or Peter with any questions. Thank you very much, Peter, and to all of you for your attention and time today.
Thank you.